The next speaker is Subhojit Roy. He just joined the University of Wisconsin. He's a professor of pathology and is the co-leader, one of the co-leaders at the Wisconsin Alzheimer's Disease Research Center. And his talk is neurobiology of aging and dementia, and I believe is focused on APP-based one trafficking and something else.
Sounds good. Thank you all. Can you hear me?
Yep.
Thank you all. Thanks to Sanjay and the organizers for inviting me. I'm delighted to talk to you today about some of the work that my lab has been doing for a little bit, and I want to show you how one thing led to another, and we have, sorry, is it not on?
Make sure you have your phone safe.
Everybody nodded when I said, can you hear me?
Can you hear me now? Is it good? All right, sorry. I want to show you the chain of events that led to some of the new data, sort of a new path that my lab has gone into, and the serendipity of it all. So this is going to be, some is going to be published data, and towards the end of the talk, about half of it is going to be unpublished data. So this is a very provocative title, I thought, Alzheimer's disease, a bottom-up approach. So what do I really mean by that? And this is, when I look at the history of how things progressed in the field, I find that there's been these important phases of research. When we started first, we had the human neuropathology, and if you go back and look at the papers there, they talk about all kinds of different things in the human brain. So you look at the human brain, that's the template for the disease, and Alois Alzheimer's described the pathology, and everyone focuses on the plaques and tangles, but actually there's a lot of things that people described in those days. You had these axons here, you have the dendrites here, they don't look good, there's some tangling and twisting and this, that, and the other, and we didn't really know which ones were important. And then came the mouse models, which has enormously helped us by understanding which of these pathologies that we saw in the human were important. And as you heard from the previous talk, mouse models are obviously great to advance the disease, advance our understanding of the disease, and they will continue to be so. But when I started thinking about neurodegeneration and looking at the mouse models, I felt that there was also a weakness with the mouse models, is that often it's really difficult to pinpoint the mechanisms that underlie the phenotype. So you can create a mouse with a phenotype, but how do you really get to the cell biology and the molecular biology of exactly what's happening on the pathways? It's difficult, sometimes impossible, and the answers that you get, even if you do get some answers, they're often ambiguous and you're not sure. So I argue that we need to get into a third phase, which is basically a phase where we can develop precise cellular models that replicate key functions of the disease, and then work with those models. But this model also has a problem, which is that often the answers you get are too simplistic. So I think this has to be an iterative process where you build these model systems, you get answers, and then you check them in mouse and human brains. And today I'm going to give you an example from my lab where we have tried to do that. And so this is what I mean by bottom-up approach. So Alzheimer's disease needs no introduction. You have the normal brain, and this is a severely atrophic brain. So I'm a neuropathologist, so these are actually brains that I did see. And one of the key pathologies of Alzheimer's, is the amyloid plaque. And it's always amazed me is that you can go to an Alzheimer's brain and take a section from any part of the cortex, and this is pretty much what you would see. You can see that the brain is studded with A-beta plaques, and this is a key pathology of Alzheimer's. Many think is the driving force. You cannot get Alzheimer's disease without plaques, we know that. And the way the plaque is made is by this transmembrane protein, single-pass transmembrane protein, which is the amyloid precursor protein. And this pathway is complicated, but basically there's three members here that you need to know for this talk. One is APP, and the APP is cleaved by a couple of enzymes. The first one is base 1, and the second one is gamma secretase. And this eventually creates this A-beta fragments that's hydrophobic and deposits in the brain as the amyloid plaque. If you look at this diagram, you can see that this cutting of APP by base is the first step in the process, and actually it's the rate limiting step. If this does not happen, then none of the downstream things will happen. So this interested me a lot, and I started to think about how this is, this is like the Big Bang for Alzheimer's, if you will, where you have the APP and you have the base, and I like to think like in blocks. So if you imagine there's two proteins, one is the substrate and the other is the enzyme, is coming together and meeting, and then you have the base kind of physically cutting the APP, and this process will eventually lead to the deposition of A beta. And that led me to think about precise experiments where we can actually study the trafficking of APP and base. And when we started this, the field was kind of very ambiguous, because a part of the problem was that most of the studies that are looked at APP and base trafficking were not done in neurons. And my lab works with single cell models. So this is one model we work with a lot. This is the hippocampal cell in culture. And there is almost 30 years of work defining how the system develops. And you have a cell that's round, and it gives out several processes. All of them, but one becomes dendrites, and then one extends into the axons. And then this grows and gives you a very predictable cell biology that you can study. So what we did was we looked at the trafficking of APP and base in these dendrites. And here you see this is dendrite, and you can see the dendritic spines here, which is transfected with this APP that's labeled, and you can see these vesicles moving back and forth. And we looked at both APP and base in the same neuron, and this was our first surprise, which is what we saw, that they were moving in different compartments. So these are things called chymographs. This is just like if a snail walks on the sand, it leaves a trail, and these trails are essentially the vesicles that are carrying APP. And these are vesicles that are carrying base, and these two are from the same dendrite. And you can clearly see that there is hardly any overlap between these two, and they are traveling in different vesicles. So this was unknown at that time, but kind of makes sense in retrospect, because you do have to keep the enzyme and the substrate separate, or everybody in the room will get Alzheimer's disease. And you can, we can't do the experiment in the human brain because there's no volunteers to give us the cells. But what we could do, if you do just biochemically separate the brain, and then you just lay it down on what is called a sucrose gradient, where you can separate proteins based on density. The APP floats on the top and base sinks down, and this is true for mouse brains or human brains, as you can see. So we think that there is this physical separation of APP and base going on. And then we did a bunch of experiments, because it was kind of controversial at that time, so we did a bunch of experiments to find out which were the precise vesicles that APP and base were moving with. And at that time, there was this view, which I think is still a lot of people hold it, that base moves in these organelles called early endosomes. And we found that that was absolutely not the case. In neurons, base actually moved in a compartment known as recycling endosomes. And there are some nuances which make it important, which I don't have time to go into. But APP actually did not move in the recycling endosomes. And APP moved, on the other hand, in this different set of organelles, which were these Golgi-derived vesicles that were going into the dendrites. Bottom line is we came up with this model based on our experiments, where after synthesis in the cell body, APP and base were sorted out into different vesicles, and they moved around in the neuron. So we are still studying this, and obviously the question in everyone's mind is, why do we get Alzheimer's disease? Are you going to get the answer in the next slide? Not really. But one interesting thing we saw was that if you remember that density gradient experiment I showed you, where if you look at normal brains, you have a separation of APP, which is here, and base, which is sinks here. In Alzheimer's, there is a striking redistribution of APP to these compartments that also has base. So maybe there is some sort of a trafficking abnormality underlying the disease, but we haven't quite figured that out yet. But these experiments led us to think about a different idea that led us into a line of research that I'm excited to tell you about. What is it? So I started thinking about this physical interaction of APP and base, and how these two molecules have to come together physically to create the A beta. And at that time, there were many different tools coming out to study protein-protein interactions. And one of them was this model called a fluorescence complementation assay. And the concept in this assay is that you may have heard of GFP. So GFP is just a fluorescent molecule. If you take GFP, in this case it's Venus, but it's the same thing, it just folds quickly, and you break the Venus molecule into two halves, then each of these halves is non-fluorescence, so you won't see anything. But if you tag it to two proteins that are supposed to interact, then if and when the two proteins interact, the molecule comes together and becomes fluorescent. So in other words, you can actually have an assay where you can see the physical interaction of two proteins. And we used it for APP and base. And the concept here is that you add, we tag the half of the Venus to APP and the other half of the Venus to base. And then when they came together, they created a fluorescent glow. I don't have time to describe all the data to you, but suffice it to say that we did see fluorescence complementation, and it was mainly it was all over the neuron, in dendrites as well as in the axons, but highly enriched in recycling domains. But turns out that the most interesting thing in this experiment was the control that we used. Now, if you think about this assay, one of the problems is that you can have non-specific interaction of these two fluorophores without the proteins actually coming together. And the best way to test it is to actually make a mutation in one of these proteins and ask if you can actually block the interaction. So if a mutation in the protein blocks the fluorescence, then you know that the interaction has to be due to the meeting of the two proteins. So we looked into the literature and we found an old paper by Martin Citron and Denis Selko, where they had made a small clip in the C-terminus of APP. So this is APP, this is the N-terminus, this is the C-terminus. What they had made is a protein called a stop 40, and they called it stop 40 because it's on the A beta 40 side. This is away from the beta side cleaving side, but you just clip a little bit of the C-terminus, and turns out that they had shown that it does not make any A beta. And what we found is that if you do that, you actually prevent the physical interaction of APP and base. And keep this protein in mind, stop 40 beta, because I'm going to come to it in a little bit. But I want to show you, just give you a glimpse of this data, that you do have physical interaction of APP and base, mainly in these recycling zones such as the tip of dendritic spines, presynaptic boutons, and so on and so forth. And as we were working on this interesting paper came out in which people looked at these Icelandic population that don't really get Alzheimer's disease. And they found that there was this mutation in APP, which is the protein that we were looking at, which actually seems to prevent the population from getting the disease. And this is called the Icelandic mutation. So we had made this assay anyway, so we thought, what the heck, let's just make this mutation and see what happens. And turns out that if you have the mutation, there is a dramatic attenuation of the APP and base convergence. And so this also kind of confirmed, at least laid some of the doubts to rest that this assay was really working and showing what we want to see. And here I'm going to show you, this is the beta CTF. You can think about this as amyloid beta. Basically, this is the precursor to amyloid beta. And essentially, this is absent when you have the Icelandic mutation, as you can see here, or at very low levels, I shouldn't say absent. So in ongoing work, we are looking at, we are doing a CRISPR-Cas9 whole genome screen to kind of find out all the genes that are involved in this process before APP and base meet, and also small molecules. But I want to talk to you about this unexpected offshoot that I've been referring to for a while but not telling you. And this is the C-terminus of APP. So I got really interested in the C-terminus of APP because if you think about it, it's kind of the stop 40 results are so odd. So this is APP drawn to scale. And you can see this is this gigantic N-terminus going on, and then you have this beta side. This is where base cleaves. This is actually the membrane itself, and this is the transmembrane domain. So really where the stop 40 is working is right here. So you can clip off this very small portion of the protein, and for some reason you completely block A beta. You completely block this protein from meeting base, right? So I thought it was fascinating, and it kind of one thing led to another, and I I thought that, okay, all of us have this wild type APP protein in our heads right now, right? Now, if by some way you can convert this wild type protein to something like the APP stop 40 protein, right, then you essentially cannot get the A beta pathology and you cannot get Alzheimer's disease. And this is the thought that, as I was kind of thinking about it, my wife, who's not a scientist, she's a physician, started bugging me about, why are you not working on CRISPRs? Everybody's working on CRISPRs. I said, okay, fine, I didn't know anything about CRISPRs, but why not put a CRISPR on it, right? If you like it, why not put a CRISPR on it? And if you think about these CRISPRs as this ideal tool by which you can go in, and if I can cut your APP in a way that it cannot meet base, then you won't get Alzheimer's disease and you won't get A beta. So I had to learn what CRISPRs were. So I just want to briefly, I don't know too much, but I'll tell you what I know. Basically, this is a genetic tool that works because of this enzyme called Cas9, which is a nuclease enzyme that goes and cuts DNA. And the way it cuts DNA is it is recognized by another small piece of RNA, which is called the guide RNA. So wherever the guide RNA is going, the Cas9 is going to go and cut the DNA. And the way the guide RNA works, it recognizes these sequences in the genome, which are called PAM sequences. So these PAM sequences are just random things scattered all over the genome. But if you can find a PAM sequence, and if you can find you make a guide RNA that goes and attaches to the PAM sequence, the guide RNA brings this Cas9 guy along, and the Cas9 cuts the DNA just few bases upstream of the PAM sequence. This is how it works. All right, so we thought that, okay, let's see if it would work with APP. So we looked at the APP C-terminus carefully, and there's these two PAM sequences. One of them, this is a very strong sequence. This is a weak PAM sequence. This gray area is the transmembrane domain. And this is very close to the stop 40 site, which is right there somewhere, right here. All right, so we made the CRISPRs. We call them CRISPR number 1 and CRISPR number 2, like thing 1 and thing 2. And the experiment is pretty simple, as you can imagine. You just throw these CRISPRs, you wait for a few days, and then you just look at endogenous. So remember, you're actually cutting the endogenous APP, and you ask if the APP has been cut or not. And the results are just incredible. So I first didn't believe it when I saw this blot. I thought there was nothing here. I forgot to load something. But this is the second CRISPR, the CRISPR number 2 that I showed you. The number 1 doesn't work. And this is the beta CTF fragment, which basically is the fragment that gives rise to A beta. And this is the same gel as here. It's enhanced to show you that there is something here, but it's the dramatic, almost a dramatic reduction in the A beta. And it's not because the protein is not being produced, partly because there is tubulin here, as you can see, but also if you blot with the N-terminus antibody, then it is there. The APP is there. It's just that it's cut. It also works in neurons. So these are again mouse neurons. The vector that we have, the Cas9, actually has an RFP in it, so we know which cell has it. There are actually 2 cells here. Oh, I forgot. One thing I need to point out is that we have an antibody that recognizes the APP C-terminus, so that if and when the CRISPR works, this portion will not exist and your antibody will not see the APP. I'm going back, sorry. There are 2 cells here. This is the APP C-terminus staining. You can see that this cell is bright and this cell has nothing in it. And this is the cell that actually has the CRISPR and the Cas9 in it, and this one doesn't. This is just the quantification in neurons, so it's working in mouse neurons. So we thought, when we looked at the sequences of the mouse and human APP around this PAM sequence, they are really similar. There's only 2 base pair difference between the cut sites. And what amazed us is that the mouse guide RNA actually did not work for the human, which in a sense is good because it speaks for the specificity of the sequence requirements for these. But anyway, we made a human-specific guide RNA, and that also pretty much knocks out the A beta. And this is also works in cells, as I showed you before. This is just genomic sequencing, just to find out what exactly it is doing. And these are the base pairs that's around the C-terminus, and this is just matching how much these base pairs match to wild type sequence. And you can see that this is the predicted cut site, and this is exactly where you have a very low matching, and the rest of the genome you have high matching because that's not cut. Same with mouse and humans, and these are the predicted sites. And for the human, if you focus on this here, it actually creates a stop codon exactly where the guide RNA is cutting. So I was told that we got very lucky, but I'm excited about these results. So again, remember we are cutting the endogenous proteins. So the way this is working is that you have the APP there, the endogenous APP, and you have the CRISPR, the guide RNA comes and cuts the endogenous APP in such a way that it cannot meet base anymore. So we call it the cut and silence approach. And if you think about it, in principle, you can probably use it for other neurodegenerative diseases as well, tau, TDP43, whatever, as long as you can recognize the region that is small enough that you can cut and kind of get away with it but prevent pathology. So the cut and silence approach, I think you're going to hear more about it, but you heard it here first. Remember that? So coming back to the APP molecule, I want to show you like what we are actually cutting is this little tiny portion of the C-terminus here. And we've done a lot of assays to look at axonal growth, synaptic maturation, so on and so forth. And so far we haven't seen any deleterious effect of creating this deletion in this protein. And as you probably know, look at the N-terminus. The N-terminus is really important in axonal growth signaling and so on and so forth. And there's been many studies on it. So we hope that by just deleting the C-terminus, we are not affecting the APP molecule too much. We're also doing this collaboration where we are making these nanocapsules in which we are putting in the guide RNA that we made along with Cas9. So these are actually protein capsules. And we want to deliver it to the human brain because if you really think about it, if you were able to actually throw in these guide RNAs in the right place at the right time, they have to work. So the question is whether we'll be able to do it or not. So this is a collaboration with Sarah and Chris at the WID, which we just started. And I can declare it now because it was released today. We got one of the UW 2020 grants to work on it, so we are excited to carry this idea forward and expand it further. So to summarize, what I showed you today was it started with just looking at the cell biology of APP and base and kind of where's Waldo kind of a thing, who goes where. And this has led to this new way of kind of therapy, if you will. And no therapy is possible, as Ron said, but hopefully delaying the onset of the disease by using these CRISPR-Cas9 based tools to cut and silence the bad guys, if you will, in neurodegeneration. And this is my UCSD lab. This is my Madison lab. The work was started with Utpal, and now a really talented postdoc in my lab, Ju Chao, has carried on the work. And one person I want to thank in the audience is Eddie, who, I started talking about APP with him, and we've spoken a lot and kind of encouraged me and motivated me to pursue this line of research. And thank you for your attention.